The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 13th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcast, The Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, The Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian Weil, and I've been in the flow cytometry space for over 12 years.
Additionally, we have a special guest today, Amrutesh. Did you want to introduce yourself?
Hey there, this is Amrutesh from New York. Been in the world of flow cytometry for about 15 plus years. Nice to meet you.
Thank you. Nice to meet you too. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with an additional location in Europe and a partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. Our guest is in New York, as mentioned, and so we're truly all over the map. We're elated to have you listening to our 13th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find The Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off for episode 13, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We do have some really interesting, some very interesting news today. So one of them is the Nobel Prize for the AlphaFold developers in the medical research news category. And here I've got an Endpoints news story linked in here where we're talking about this is the first sort of AI-related Nobel Prize, if you will, for this segment of it, I suppose, in chemistry.
Right, and you say the first. I know we've been talking about AI a whole lot. Certainly very interesting, constantly evolving. I guess that's what AI does, right? So, but anyway, with this, it was really interesting because the first time I heard about this was through my cousin who actually works for Google and was talking this up, being associated with DeepMind and whatnot. And yeah, really looking at predicting the structures, the three-dimensional structures of proteins, and aiming to get above what had recently been a 40% accuracy threshold. And at the first attempt had gone over 60% and has since improved upon that. And it's important because there are so many protein sequences that are known, but to the point as of 2021, there were only about 100,000 protein structures known. And historically, a lot of the structures were determined by X-ray crystallography, which you would imagine it takes quite some time to be able to experimentally deduce something like that. Whereas with AI, if you're able to predict it with a high level of certainty, you can get through a lot more understanding a structure, which then follows you're able to understand drug design, any kind of interactions or potential side effects that could be had from therapeutics based on this kind of knowledge. So really cool to see this come into the fore and really how quickly they were recognized for this Nobel Prize.
Absolutely. And I don't know if we ever talked about it. My first research experience was in protein crystallography. So I had to grow the protein and tag it so that we could purify it and then purify it and try to get it concentrated enough for crystallography. And, this is probably coming from an undergrad's research perspective, but it's just a terrible process. My goodness, the amount of work required. So very excited about this. And, yes, it's good at predicting sort of known things and things that are possible via standard crystallography techniques. But the exciting thing, too, is that this could be then applied to all the things that we can't do, right? Protein crystallography assumes that you can express, purify a conformation of the protein that's physiologically relevant. For all the proteins that have sections that are not amenable to crystallography, this would be a huge jump, because the first step of crystallography is always known as black magic, where you just find a condition that works and you try like hundreds of or thousands of different conditions to get the protein to crystallize. So if you no longer have to go through that step to get a reasonably accurate thing that you could experimentally verify, that'd be a huge win for the field. So I can see why they got it. And hopefully we'll see some of the outcomes of that in the near future.
I don't want to sell crystallography short, though, because some of the coolest images definitely come from those crystal shots. Always liked it. And I'm not just talking about that as kind of fluff. I mean, there's so much information that's gained from that. But you're right, it's a process, right?
Yeah.
And interestingly here, one of the things they talk about was, it's easier to predict something if you have homologous proteins, homologous structures to base it off of. But here, what they're working on as well is when they don't have something like that, to be able to really still nail the prediction.
Yeah. Incredible steps moving forward. So, very optimistic there. And then the next piece of medical research news was just a very small paper about immune signatures and being able to use sort of a wider immune signature to predict adverse events in immunotherapy.
Yeah, we always talk about checkpoint inhibitors, right? And here, kind of coming to the fore, continuing again to be able to identify whether or not the presence of these inhibitors will actually lead to being able to predict adverse events. Specifically then looking at increases in T cells, your Th2s, your Th17s, as well as specific cytokines that could portend adverse events. And so certainly makes a lot of sense and really interesting to try to gather this information and use it as that predictive tool.
That's it. And, the immune system is so complicated, it's difficult to figure out what that looks like against some sort of either reference immunome or reference immune system function. But definitely this paper and this article that sort of links to the paper made a good stab at it. So hopefully we see more and more of that sort of thing at the front. Then you can start to see the potential for things like Cytec panels as screening parameters or other things for clinical trials.
Yeah, so these panels are getting so much bigger and bigger, and isn't that's the topic of our discussion? Oh wait, we're going the other way today, aren't we?
We are indeed. Yeah. So exciting things in the future of immunotherapy there, medically at least. And then investment-wise, the two stories I had were both local Philadelphia stories. So the one is on Jim Wilson and his new spin out from UPenn.
Yeah, exciting to see the growth of the industry here in the Pennsylvania area, the Philadelphia area, spinning out of UPenn, Gemma Biotherapeutics, which essentially looking at rare disease research, launched October 1st of this year. And then pairing with that, Franklin Biolab, so a CRO for gene therapy. But essentially the two companies they're distinct entities, but working together.
And it's interesting because, of course, gene therapy has been on a pretty significant downturn for the past year or two. The UPenn gene therapy program was definitely a trailblazer in the field. So it's interesting to see that this is splitting out into a private company now, like during a downturn, because we think that there is, or Jim Wilson at least thinks that there is additional opportunity there that's not being able to sort of be taken advantage of.
Yeah, and gene therapy. It's there's a lot to it, right? I know I've had personal experience. My nephew was went through a gene therapy clinical trial, and increased his quality of life significantly, and frankly his longevity too. And so know that the technology holds great promise, but there's always things that you need to investigate to make sure that it's working properly and essentially getting to the larger portion of the population that needs it.
That's it. And, the question in gene therapy has shifted from one of can we do it to can we afford to do it?
Hmm.
So that is usually a question that is answered quite well in industry as opposed to academia. So it'll be interesting to see how these companies fare, and interesting to see how that works out for the Philadelphia biotech ecosystem. So
And that slip was certainly no judgment on your part.
it is true. It is true. Yeah, and the other investment news is on Tolerance Bio. So that is a new biotech that's launching in the Philadelphia area with some prevention veterans.
Yeah, so looking at the thymus, right? And this is essentially where your T cells come from as far as gaining tolerance. And so I thought of as the master regulator of tolerance. What that really means is the T cells are primed to not recognize self, right? Otherwise, you have issues. If the T cells recognize your own tissues, then your body attacks your own tissues. And this is where autoimmune disorders come from. And so, trying to make sure that you've got that appropriate balance. And so, here they're looking at an allogenic iPSC, so off the shelf essentially, that could be infused. Now, the iPSC is an induced pluripotent stem cell, and so basically can direct the fate of this cell into different directions, whether it be an NK cell or whatnot, and then generate a therapy. And here, I guess, let's see. Yeah, like I said, looking at immune mediated diseases.
And it's interesting, and definitely the allogenic approach is the answer to that question we just posed. Can we afford to do some of these crazy things that we've found that we're able to do? So allogeneic is the best process for that. That really decreases lead times, so that their therapies on the shelf ready to go, and can then be quickly deployed, and then also the cost is just significantly less than personalizing it for each person. So by those mechanisms, very good. But for the iPSC, that's still a relatively difficult route to go for different companies. So we'll be looking forward to seeing how this develops as well. They mentioned all the appropriate targets there, like cancer, autoimmunity. So will be very interesting and good as well to see this as a positive sign for just overall biotech investment, that people are still willing to go out and take these relative risks on some new therapy and new directions here.
Right, and talking to exactly why tolerance is looking this direction. So the thymus function reduces as we age, right? And so you have a loss of self-tolerance. And essentially you've got naive T cells circulating, but essentially as you age, they become fewer and fewer. One of the things that tends to do also is reduces your ability to get a benefit from things like vaccines. And so that's why we talk about the elderly being more need for protection there when we talk about was it herd immunity, right? And so here this would help to restore the function of the thymus and hopefully extend the ability there for naive T cells circulating.
Yeah, we'll look out when they start announcing targets. Excellent. All right. And then in terms of KCAS news, I put you and I on there, Adam.
You did, but I got to be honest. I think yours is mine pales in comparison to yours as far as our travels are concerned. What Brian's talking about, we've been going to conferences, visiting sites, visiting clients, and so for myself, I was out on the West Coast about a week and change ago. First at the ICCS conference, really great to connect with various clients, potential clients, vendors, competitors. It's kind of one big family out there, and it's a great excuse to get together. And then from there, I went down to San Francisco to visit a number of clients before coming home on the red eye and going to work that next day. Now, I would say, Brian, why don't you regale of all your travels here?
Yeah, I mean, historically, in October, I started out at our Lyon, France lab, of course, to look at the two new lovely SciTech instruments and talk to the group about how we're working together on some new global trials on them. So that went swimmingly. Then went over to the Festival of Biologics in Switzerland. And that was really interesting, and where I got the source of the paper that we'll talk about in just a moment. But really exciting to see all the biologics being developed and how the European ecosystem views them and the biotech industry as a whole. So that was excellent to connect with clients and peers and things like that there. Of course, staying here in Philadelphia for a couple days this week, and then heading off to first Los Angeles, and then the Australian Biotech Days down in Melbourne, Australia next week.
It is nice to have you here for a couple of days. Absolutely. It had been quite a while since you were here. But sounds like a lot of useful time traveling there.
It is. And, it really makes me think about why we go to these conferences. And one thing, again, from my early undergrad days is that my professor that worked with protein crystallography was always adamant that we think about why we were doing the experiments that we're doing. Like, why are you trying to crystallize this protein? Who cares? Because his worst fear is something that happened to one of his colleagues, where they looked up at their career after 20 years and realized that they had just been playing with rat pee for 20 years. And that some of the proteins that they were working to crystallize weren't really a good sort of translational targets, or didn't speak to some of the underlying questions that they were looking at. And you never want to look up and realize that you've just been playing around with rat pee for 20 years. So going to conferences, connecting with our peers, make sure that the whole field is aligned. What are the challenges? How is what we're working on today going to address some of those challenges and lead to better outcomes for patients moving forward? So
Absolutely, and it's interesting. So with all the travel, yeah, you'll be tired, but the consolation is that you do make these connections. There were great workshops at ICCS, and talking about opportunities for future workshops and what that might look like. In this case, it doesn't sound like there was much consolation in getting to work with rat pee for however many years. But everybody's got their own thing, I guess.
I hope that there was some sort of other silver lining to that story, but I didn't get much more than that, other than my professor's warning, like, don't do that. So
it's good warning for sure.
Indeed. But that does lead directly into, of course, the paper for this month, which is the paper from Chris Klein and company about the present and future of bispecific antibodies for cancer therapy. And the reason I picked out this paper for this month was, one of the people at the Festival of Biologics in Switzerland was Chris Klein, and he basically gave an overview of some of his research. He was previously, I believe, at Roche, and has since moved on to more of a sort of biotech incubator type role. And so as part of that, he had to give his talk on sort of published papers, and this was where he spent the most time. And we were talking earlier, Adam, and I was saying that I think that it should be almost a requirement to have some sort of recorded narration of these review articles, because his talk was just wonderful in terms of sort of where the field was, sort of how it got to the point that it is today, some of the big developments, and where he thinks it's going in the near future.
Yeah, and to your point, I mean, it is something where it's a big paper. There's a lot of information in there, and when I say big, it's just really helpful to reference there. But yeah, the narration, then you get an idea of really where the author felt should be the focus and what you should take away from it. I will back up and say, this is a Nature article, and so we typically mention about the impact factor. Nature is a very well-known publication, and the impact factor is 50.5, so the highest of any of the ones that we've reviewed so far. That was as of 2023. And yeah, talking about bispecifics, we've mentioned this a couple of times. I think just last two podcasts, even, we've had conversations here, and one of the bents on it was just that it's emerging, it's a newer technology, and you and I had talked about the fact that we've had experience with this 10, 15 years now, but that's in development, right? And that doesn't necessarily mean that it's got the light of day shining on it as far as outside the industry is concerned. Really cool is the synergy between bispecifics and chimeric antigen receptors, in that you could think of a chimeric antigen receptor cell where you have a targeting arm, and then you're bringing that T cell to the tumor with that targeting arm, but then internally it gets activated, and essentially that's very similar to a bispecific where it's got two functions. So there are a couple of different types, right? You've got your combinatorial and your obligate. So the combinatorial, as it basically implies, is that it combines the effects of two monoclonal antibodies into one bispecific, whereas the obligate is two arms. They're required to both bind to be able to get some sort of function. And so here we'll be talking about binding a T cell on one arm and a tumor on the other to bring the two together so that the T cell can kill the tumor cell. And there's a lot of different ways that you can go about generating a bispecific, and the engineering too specifically can be very challenging, because in nature you have two arms that just bind to themselves for a monoclonal antibody, whereas in this, what you're trying to do is take one arm from one monoclonal antibody and add it to another arm from another monoclonal antibody without having them just homodimerize, right? And so here you want that heterodimer. And so there's techniques, knob and hole, things of that nature to be able to achieve that.
That's it. Yeah, and definitely the stabilization of novel combinations of these antibody structures, but then also antibody functions, so that you can have one arm doing one thing and then the other arm then responsible for sort of an entirely different activity. Was definitely a focus of the conference and a focus of Chris's talk as well as his paper.
What's cool too is we talk about bispecific here, but there's multi-specific, and some of the engineered versions that I've seen where you've got one arm, you got the other arm, but then you've got the FC region, and then you've got multi-bodies on the back that do things. It's just incredible the amount of work that goes into it to be able to figure out how to get something like this to work. And it all boils down to a little piece of engineering for protein engineering that I was found interesting is the glycine linker. And what it does is essentially you have a sequence of glycines and serines that impart flexibility. And a lot of the papers I've read talk about when they're putting these kind of bispecifics together, utilizing something that's just as simple as that, but it imparts that flexibility to allow these bispecifics to function. Now, obviously, there's a lot more to it, modifications that alter half-life so that they don't just fall apart or they don't just get recycled, and making sure that you don't have certain toxicities, and so humanizing if it was brought up in mouse or rat or something like that.
That's it. Yeah, I mean, each one of these sort of advances, right, takes 5, 10, 15 years to figure out, okay, here's this problem. how do we solve this specific problem in this context, and then what can we generalize from that, and then spin out a company, get a patent, and now the industry can use it. And it's just that over and over again, which again speaks to the value of these conferences, where we can all talk about which problems we're looking to address and how we can sort of iterate and move forward there.
Yeah, and so you can tell I did a lot of work on bispecifics and antibodies in the past, in previous jobs. I was interested to see that there are more than 300 clinical trials ongoing for bispecifics, on more than 200 molecules. And so really, a lot of these are coming out. Now, there's not a ton of them that are approved just yet, but they're in the testing phases and hopefully will be coming along. Now, there's, a large portion that are hematological, meaning within the blood versus solid tumor. A lot of new ones are aiming on solid tumors, so bringing the T cells in proximity with the tumors, in that case, to destroy a solid tumor. But yeah, it definitely holds a lot of promise. And it's similar to, at the outset of this, we talked about chimeric antigen receptors. There's a lot of engineering that goes into that. And the hope is that that becomes an off-the-shelf therapy. Well, bispecifics are looking to do very similar and have a lot of engineering challenges themselves.
Sure. And they do share still the propensity towards cytokine release syndrome, right? So that's always a primary consideration when designing any of these therapies. How do we make sure that we don't overstimulate the immune system and generate sort of a runaway response?
Yeah, absolutely. And I was listening through just in this paper all the different options that are available. Different, I shouldn't say options, right? But mechanism of action that people are utilizing specifically for it. And one of my favorite ones is blocking two ligands. I kind of think of it as like a paper towel to clean up stuff, and I'm going to have to share stuff. Those for sensitive ears maybe just want to pause for a second or two or skip over. But this morning I get into the kitchen and there's a Post-it note on the cabinet. My daughter had left and there's an arrow pointing down and it says, "Cat threw up." And I had just somehow stepped over this large pile of cat throw up because my cats had decided to open up a bag of hamburger buns and eat hamburger buns, which I didn't know cats did. And my daughter just left the Post-it to warn that was there instead of cleaning it up. And so that's where I'm saying this, maybe it would have been nice to have this blocking two ligands antibody could have cleaned that right up. I don't know, or I'm just totally going off a far afield here.
Yeah, and just to reiterate to our listeners, we are in the paper of the month section where we are talking about bispecific antibodies. This is not yet the banter section. We're getting there though, so no worries.
I think it was applicable though.
I agree wholeheartedly. Like the functions of these things, like you can describe in layman's terms the function of these things, which is one of the impressive parts about these review papers and this review paper specifically, that when you break down what the antibodies are doing, it's pretty straightforward, right? Like you want it to bind to one spot and bind to another, or in that case, block everything, or stimulate something else. And it's just the engineering that goes into enabling these different functions that is the exciting part there.
You know what it is? I didn't have a FC function for her. She knew that there was cat puke there. She didn't know what activity to do next. And so had she had that portion on her antibody, she would have been able to clean it up herself.
There we go. Improvements all around, which is probably a good segue, or I don't know, just a good time at least to talk about our main portion of the podcast today. So for this main portion, we are talking about when can a smaller panel be desirable over a larger panel. And as we sort of introduced earlier, we've got a special guest with us, Amrutesh. And Amrutesh, if you could give maybe a little bit more background on yourself and what you do on a regular basis, then we can jump in.
Hey everyone, this is Amrutesh Puranik, and I'm the founder of Bespoke MultiOmics Consultants, and I do consulting. My basic training has been in macrophage biology and nephrology and aging. Later on, because of my love and ability to do these large panels, transitioned into flow cytometry that has been always a core of my research. So with that, came to trained at Mayo Clinic in Minnesota, came to NYU, then moved to a biopharma world and been in biopharma since then. Uh, been a flower at heart. You could call a cyto geek, a flow geek. Like to design panels, spend hours looking at those colorful dot plots, which we call as tarot cards. And in my free time, I like to DJ, which means generate a panel and keep tweaking a panel so that I get something like a perfectly optimized unmixing matrix. So yeah, that's the summary of what I do. But been a flower at heart for about 15 plus years.
Oh, exciting. And, this is the first time you and I are talking. Really excited to have you on our podcast. And also, too, you mentioned about having studied macrophages. That was a focus of my PhD research. So, really excited to have you on as far as that's concerned. And so, Brian had mentioned about our topic here, looking at when it's preferable to do a smaller panel rather than a larger panel. Obviously, the industry is moving more and more towards larger panels, getting more and more information. And I think the first ones that we can dispense of right away for when it might be favorable to do smaller panels would be minimizing cost, right? If you don't have a ton of resources behind being able to do it, it's certainly cheaper to do a smaller panel. And then also, if your instrumentation can't handle doing a larger panel, then obviously it's beneficial to go in that direction. But curious to your experience and thoughts on, you mentioned about trying to optimize the larger panels and get to that perfect unmixing. How about for smaller panels? Some of your thoughts there on when it would be preferable.
Pardon my sarcasm, but the answer is never. Uh, been studied flow, learned flow in early 2000s, and later on been practicing flow for 15 plus years. Started from Cantos to LSR to Fortresas. Uh, uh, and things have evolved over a period of time, and small is or large is a very subjective term. To different people, it would mean different things. Even in our world of flow cytometry, 15 to 20 colors is a piece of cake for some. 15 to 20 colors is a large panel for some. But at the heart of it, as an immunologist, what I like to bring out is what's your research question? What's your goal? What are you trying to achieve? So, if your goal is I want to develop and study receptor occupancy, and I know you guys have had an entire podcast focused on something like an RO. So, an RO is a very specific goal. It's a very specific set of assay. It has a regulatory binding to it. So when you have a very focused question which can answer your uh, biological question, then yes, a smaller panel is very desirable. It's very effective. It can give you the answers what you are asking for. Certainly, besides the cost, I think reproducibility is also a bigger consideration. Can you reproduce what you're trying to do? Um, and then, of course, we can have some targeted insights into uh, specific biomarkers. I do a lot of biomarker work, and for the most part, if it's a trial-related biomarker, then it very quickly becomes an exploratory study rather than a focus study. So when it comes to asking focused questions, smaller panels are definitely better.
It's true, and in general, it's then easy to get the answer to that question in a very defined way, right? Where if you're collecting lots of information, I'm reminded, of course, of 23andMe, the sort of consumer genetic testing company that did a lot of sequencing on a lot of different genomes, but then when they went to try to sell that information to pharma companies, it wasn't done in a way that would have been consistent with trials, from my understanding, so that it couldn't then be used for that. So the concern is, yes, if you run a large panel, you might have something that you didn't expect and would be able to capitalize on that. But then you might also find that you didn't run the assay in a way that allows you to answer questions about sort of those other markers. So if you have this sort of plan for how you're going to interpret the data, you can make sure that the study is powered sufficiently to answer your questions there.
That's right. Absolutely right.
Now, I think another consideration might be the team that's going to be performing this, right? So if you've got a team that may not have experience on larger panels, it would be beneficial for everyone if it were shrunk down a little bit, right? You're not looking at as many markers, you're not having to deal with as much in the way of spillover, compensation, things of that nature. To your point, it goes along with the RO. You really want to focus. Now, RO itself can be somewhat complex assay, but if we're taking a step back, we're doing an IP, but with a more green team, then shrinking down the panel might also be beneficial.
That's correct. Again, so we start with what's your biological question? What's an absolutely essential markers to your panel? What are must-haves? And then what are these we would like to have? So when you go step by step, starting from a biological question, is your biological question satisfied with a 10 color panel? Probably that's what you do.
Mm-hmm.
If your biological question needs a 20 color panel, that's what you do. Have 20 biomarkers. But then going from 10 to 20 is again challenging, draws a lot of other questions as on reproducibility. Do you have the capacity to draw it? Do you have the gates that are set beforehand? Have you done all the bench work? So there are a lot of these considerations which are usually uh, need to be considered when you're doing something of this sort that is clinically reproducible from one, let's say, a central lab to other central lab.
Sure. And, once you start getting into multiple lab scenarios or multiple sort of time point scenarios, you have to start thinking about what happens over the course of time, right? So if you have a focus question, then you have, let's say, 6 or 10 or 20 reagents maybe that you have to monitor for lot changes, make sure that there's no change in conjugation efficiency, make sure that you understand, again, how you're going to interpret that data. If you all of a sudden balloon out to 20, 30, 40 colors, now each one of those things, as you mentioned, Amrutesh, could have an impact on mixing or how well optimized that assay is, and may need to be sort of re-optimized every time you change a lot. That could have an impact on one of the other spectra that may overlap with. So all of a sudden, that assay becomes a lot more difficult to manage, more so than just going from 20 antibodies to 10 antibodies. Now it's also all of the lot changes and understanding what that means across the entire spectrum of the panel.
Now, taking a bit of a tangent with that, one of the things that we had seen several years ago, especially when we were really in the height of the pandemic, was availability of reagents and specifically antibodies. Consistently, every single time we were trying to design panels, there were 1, 2, 3 antibodies that really just weren't available. I'm glad to see that as a whole, industry's moved on from that as far as being able to get things generated and available, and especially quickly. Certainly not easy, but that had been a consideration too as far as panel size.
Absolutely, absolutely. Well, as you go from small panels to large panels, reproducibility, availability, and if you're having a custom conjugated antibody, how good is reproducibility in that conjugation? These are all central to deciding if this panel is viable for the analysis or not, because you can plan all sorts of analysis on your data, but if your raw data isn't reproducible, then it really becomes a challenge. If you're doing longitudinal studies, especially, these things keep showing up. You can do percentages, and that's the most reproducible thing. But every time you have run a batch, what are your controls, and can you afford to run that same controls every single time you're running an experiment? Is really critical to these types of analysis.
One of the things I was curious, you'd brought up RO assays, something that's a bit more difficult. Another one that I've seen a lot of where generally the panels are paired back is phosphorylation studies. So curious on your experience there.
Yeah, so there has been a lot of so phosphorylation has come a really long way. We used to do if we are doing fresh samples, we try to do a surface staining versus intracellular staining, and we would do two tubes, and then we get data from both tubes. One would have something like a Ki67, one would have just surface markers. Since then, since those times passed over past few years, now we are kind of moving into where we are fixing cells, and it's like you saw there have been reports on in cytometry on using a overnight staining panel. So I think these panels have higher potential to be successful when it comes to looking at phosphorylated stats, especially looking at signaling events related to, let's say, tocilizumab like biomarkers, where we clearly want to study a certain signaling downstream of, let's say, IL-6. So those panels, those methods, I definitely put my money on. And so far, we have not seen a lot of data except for that first seminal paper. But we are adopting some of those techniques of overnight staining and trying to see if we can really measure and catch something like a p-stat 5 or a p-stat 1 like protein, and different clones of these in a clinical trial setting. So yeah, I'm very positive and excited about those developments.
Excellent. Thank you. Yeah, it's certainly interesting. I had done some stat staining myself from a PhD, and we regularly do that here, the phosphorylation assays. And so, yeah, the stats always seem to be very central to those types of studies. Brian, any other thoughts as far as smaller panels on your end?
The only other thing that really comes to mind on my side is definitely the cell sorting. So the fewer markers that you can use to sort out cells, the better in most cases. Anytime you have to go internal, then cell viability starts to decrease. So trying to keep it as a surface stain, and then trying to keep it to a small number of colors, if at all possible, usually makes for more successful sorting.
Absolutely.
But always good to have sponsors come in and be able to discuss sort of what the focus is and what the research question that we're trying to answer is, and have a well thought out plan in terms of how we're going to interpret the data to really be the foundational part of this. Other than some of the impacts we talked about on cost, on reproducibility, on the number of lot-to-lot changes, and sort of the overall experimental plan that you had to think about for large panels. Any other good reasons to go with a small panel?
Yeah, certainly something like receptor occupancy or something that you want to do longitudinally, smaller panels are definitely more reproducible. You can track their changes. Uh, uh, I think you have discussed instrument changes in the past, and small panels are rather not as sensitive to these changes as compared to a 42 color or a 50 color panel these days. So certainly the flexibility and reproducibility in small panels is relatively better, especially when it comes to, again, I have done a lot of RO, so I like to bring it back to an RO. When you look at let's say, is studying a simple checkpoint inhibitor and developing a checkpoint assay or an RO assay for a checkpoint inhibitor. Uh, if you're looking at myeloid cells, are you going to block your cells? It's a very, very simple practical considerations, because blocking antibody, you are basically using an IgG, and you're going in with a secondary that also uses an IgG. So these considerations are really, really important. Not that they can't be done, but then you have to make sure that those controls are built in when you do standardization for these assays. And usually the assay development part is the amount of time given to an assay development has to be that optimal, that the assay is validated before it goes into something like a production setup.
And that certainly is another consideration you mentioned about the amount of time that it takes to get the assay optimized. And if you need to qualify, validate it, certainly larger panels can tend to take longer to be able to optimize, just with all the considerations for the interactions between the different fluorophores, different markers, so that you get it right. And that's why you even started right at the outset of saying that you really enjoy getting that perfect spillover matrix.
Yep, absolutely.
And it's
Absolutely.
not easy to do, certainly quickly, although it always asked to get it done quickly. But I mean, those things can take
Yeah.
months.
Everyone needs the standardization to be done yesterday. And then when the data starts percolating, we always have gone back to, oh, if you're blocking PD-1, let's say, why do you see PD-1 after the treatment? So there have been so many instances uh, where I do a lot of biomarker work where people have looked at, there are follicular T cells associated with the response, and we looked at follicular T cells. Oh, how did you measure follicular T cells? Oh, we used PD-1 and CXCR3, CXCR5 to identify follicular T cells. And okay, why would you think PD-1 antibody would work when they're on a checkpoint inhibitor? And lo and behold, it does not work. So a lot of these considerations are overlooked when the biological questions are set. So getting involved with the clients as early as designing these experiments when a trial is designed is always beneficial, rather than correcting it later. Uh, and then trying to find a workaround for these cells that you have set to look out.
Yeah, I think you brought up something. Biology doesn't always behave the way that you would expect, and it has certainly been in on calls as far as part of those questions. So, what happened here, and you can tell anybody on the call, listen, everything was done appropriate. We've got worksheets, we've got documentation, staff is trained perfectly on this, and so confident that everything was done right, but all of a sudden there's questions in the data, and you're right, it takes then that partnership with the client to be able to figure out, well, what sort of information is there out there to help us to understand the data that we're getting in.
Absolutely, absolutely. And then that's the reason, that's where the travel comes in. That's where you have to meet the client, and that's where you have to win the client over. And so that, before these questions come, they're already addressed. So, that's where conferences are really important, where you can meet people in a not so formal setting, and try to learn their expectations. As far as my field goes, it's the most important, like what exactly you're looking for um, when you are giving me this contract.
Absolutely. And so I know we do have a hard stop today as far as conclusion of the podcast. And so want to give you the opportunity, any final thoughts there on smaller panels or just flow in general?
Well, I think flow is getting into an exciting space. Of course, we have these questions of data analysis, what to do with the bigger panels, what are different types of analysis that we can do, exploratory versus more conventional analysis. So I think next few years are going to be really great for the world of cytometry. Uh, and Right now, a lot of people ask me to use AI or ML in Flow, and well, we use a lot of machine learning methods. The problem with the Flow is we like the GPT or in the chat GPT is the T is transformer. It's the context. Right now we have so many cell types that sometimes the context with which what we are talking about is different to different people. So I hope at some point in time we basically agree on to what are these cell types, and how can we measure these cell types, especially in the field of biomarkers, where there have been the checkpoint inhibitors have been in existence for past 10 years, but there is no single trial that tells us that, okay, if you have checkpoint inhibitors, these are the biomarkers for those checkpoint inhibitors. So I am looking forward to having some kind of consistency in these using flow cytometry.
Nice. Yeah, fully agreed there. And in fact, that's a good segue into our fan feedback request here in terms of what are the highest impact panels in the history of flow cytometry. And so that's a question that we pose out to our readers, and we'd like to get their feedback on what we think the biggest panels are. And the reason I think it's a very good sort of association with your ending question there, Amrutesh, is when I thought about it, I thought primarily about HIV and how important the CD4 to CD8 ratio is, and just how essential that very small parameter is. That's very easy to get to, and every lab in the world can now do that. But it's also completely harmonized, and a lot of the manufacturers all use the same IVD kits. So to me, that's one of the highest impact panels that's ever been developed, and why again sometimes a small panel can be as effective or even more effective than some of these large ones, and why the naming of it needs to be consistent between the field to have these types of sort of widespread impacts.
Right, getting down to the research question, right? That's what was needed to be answered for it. It does it. It's elegant. It's simple. You don't need to add anything else on.
Yeah, absolutely. All right, and so then we can move on to sort of the wrap up section. I'll bore you with one joke. I don't know how good it is, but then on to the banter. So the joke, Amrutesh, is related to just discrepancies in terms of how we interpret different cell types. And I was thinking about some FOXP3 gating recently, and came up with a joke that I told my children that they didn't think was funny at all. But
No.
it's basically saying, what is the scariest type of flow cytometry?
What's that?
Boolean gating.
Oh,
That's a good one.
That's good.
That's a good one.
Now, do they know what
man.
Boolean is?
They do not. It took about 10 minutes to explain, and then they were already playing with Legos again. So
Yeah, I'm sure. It's definitely a good attempt. I like it.
We try our best. All right. But in any case, in terms of banter, we wanted to sort of end the session with some sort of lighthearted things. So wanted to get the most interesting thing about some recent trips. So Adam, what was the best thing about the West Coast?
Oh, it was gorgeous. I will say one of the most interesting parts was coming back from the final client visit and going into San Francisco. So going from Oakland to San Francisco, going over the Bay Bridge, the Blue Angels were flying around doing formations, tricks, whatever. I think it was Shore Week was what Chris, our BD rep, was saying. And it's funny, as I'm watching that, all of a sudden, I hear in the background there's an emergency vehicle coming up, and I get this image in my head of Godzilla movies, and like all of a sudden you're stuck on a bridge and there's planes flying around and emergency vehicles, and I won't say that I fully expected Godzilla to pop up there, but I was a little bit disappointed that he didn't. With that, it was just gorgeous. I mean, San Francisco was amazing. Got to eat at a fantastic restaurant there. Seattle, just connecting up there with so many people from the industry, really appreciated that. And so, yeah, overall, I really loved that trip.
Nice. Excellent. Amrutesh, what about yourself? Any big trips that you've taken recently or in the near future?
The West Coast trips, I mean, I was in Minnesota for 8 years, so I have all great memories of the West Coast trips because I used to get out of Minnesota, and Minnesota is usually cold for about 7 months of the year, so I used to love going wherever outside. The European trips are especially challenging if you're flying over France. They I can't speak French, and we try our best to pronounce the French words, and especially when we are trying to get a croissant or something, and that's always a challenge.
Yeah.
Because they never approve of your effort, I would say, and their eyes can say a lot of things. Apart from that, Paris airport is always fun, because the way the connecting flights are scheduled is to give you a tour of Paris airport. So they make you go from one end to the other end, and that's never fun. So yeah, those are. But yeah, apart from that, it's nice to travel and meet new people. I like that.
It is nice.
How about yourself, Brian?
Yeah, I'd say the train stations in both France and Switzerland were pretty wild to me. You just get a ticket and then you get on a train, and they don't tell you where your train's going to be or anything like that. So I spend a lot of time in airports and on airplanes, and everything is just so structured. Like, you know where you're supposed to be like 3 hours ahead of time, and so on and so forth. And here it's just a lot more relaxed. So that was interesting. And then definitely the reputation for Swiss trains is well deserved. Like, those are some of the most impressive and time oriented train stations I've ever been at. So definitely a cool thing to see.
Very cool.
Yeah. And then last topic, I guess, was best practices for teaching children to drive. My 5-year-old was trying to learn Mario Kart, and I was trying to instruct her, and she wasn't having it. So I wanted to get any good takes on it from you, Adam, or Amrutesh. I don't know if you got any experience with teaching young people how to drive cars.
For me, teaching kids how to drive, especially when you're sitting in the passenger seat, it's amazing how many times you want to put your foot down on an imaginary brake pedal that it just is not there. I think when you're in the car in a certain situation like that, the most important thing is just to remain calm and not let them see just how much you're stressing out about it. When it's playing Mario Kart, I just you keep on beating them until they get better, because their competitive instincts just want to come out, and eventually it does. And then they appreciate the fact that they just beat mom or dad, and they really earned it.
Nice.
That's true. I mean, rather than teaching kids, just be with them, and especially when it comes to video games, the later they know, the better it is. So just have fun while they struggle with it. At the mercy of, they might throw the remote at you, but apart from that, they are going to master it before you know it, because they are very focused. The thing that I like about young kids is that they'll if you're laughing at them, they'll make sure that they learn these things, and then they will want to beat you. So yeah, it's kind of fun to teach them, to rather watch them learn these things on their own, and then not play with them, because that's a bit frustrating.
Then when they can beat you, how do we learn better too? So
then they're merciless too. Once they figure out that they got you beat, man, it's over with.
They're merciless, and they don't show empathy towards that.
That might be the drawback of this kind of tactic.
They're very gentle with their moms, but with dads, they're especially ruthless. So it's fun. I have an older boy and a younger girl, so I get to see all ages. It's fun.
And I will say too, Brian, going along with that, I had a game where it was something I had played since I was a teenager, and I was going up against my son, and I kept on beating him. And he was fairly competitive, but I would win most of the time. He got fed up with it at one point and decided he was going to show me one of his games. And so we started playing. I was like, okay, so what do I do? He goes, figure it out. I was like, you tell me? And he just beat the living daylights out. He was having so much fun. I was like, all right, fair enough. I guess I deserve that.
You asked for it.
Goodness. All right. Well, things to look forward to, I guess, as we start to see more and more video games and/or cars. I keep hoping for that self-driving car, so I don't have to worry about it. But I think it'll be some time. But thanks to all of our listeners for joining us. Next month we'll have an episode focused on some analyst variability. So looking at both how we can manage variability for manual processing as well as automation. So looking forward to that.
Yeah, and thank you, Amrutesh, for joining us. Really appreciate it.
Oh, thank you so much. Your podcasts are really, really helpful. I have listened to at least about half a dozen of it. So thank you so much, guys. Keep going at it. It's very important.
Thank you.
Yeah, appreciate it.
Thanks, everyone.
Bye.